Entonces, tienen plataformas para organizar los datos con la misión de que se puedan trabajar mejor y hacer colaboraciones entre colegas de todo el mundo, que sepan que tienen un formato universal con el que pueden trabajar, además de que ahora las revistas internacionales piden que los datos tengan, haya acceso a los datos con los que se trabajaron. Entonces, estas plataformas son ideales para no hacer demasiado trabajo, para hacerlo una vez y después que nuestros datos se puedan compartir, que la ciencia sea abierta. Entonces, esta es una labor muy importante. Y también hacen trabajos de optimización del flujo de datos. Esto es cómo se trabaja con los datos, visualización de datos. Trabaja con el Allen Institute en ese sentido, con el MIT, en una base de datos, una plataforma donde se pueden subir los datos neurofisiológicos de manera gratuita. Entonces, es un esfuerzo muy importante para tener más información acerca de los experimentos del cerebro. Y con los laboratorios Berkeley desarrollaron una plataforma que se llama Neuroscience Without Borders, NWB, Neurociencia sin Fronteras, que es justamente de lo que nos van a hablar hoy, de cómo manejar los datos, cómo subirlos a esa plataforma. So, Ben Ditcher, thank you very much for being here. Please, go ahead. Apague el micrófono.
Okay, thank you so much for that introduction, for inviting us here. This is the first time we've done this type of event in Latin America. So we're very excited to expand our reach. We've done a lot in the United States. And, you know, I've learned about all the great neurophysiology research being done here. And I thought, you know, this is meant to be neurodata without borders, right? Without the borders of a specific country. You know, we really want it to be a worldwide effort. So it's really great to see so much interest here. I know several of you traveled quite far to be here, and we very much appreciate that. Really excited to work with you with this intro session, and then also later today, and if possible tomorrow. Thank you so much for the wonderful introduction. From what I understood, it was a great intro. So I'm going to skip 10 slides. No, I'm just kidding. I will recap some of that. But really, thank you for the beautiful introduction. Okay, so this is Open Neurophysiology with NWB and Dandy. I'm Ben Dichter, or Benjamin, if you prefer. And this is Alberto Mayorquin, who is part of the Catalyst Neuro team as well. And Alberto will be leading some of the more in-depth sessions later today and helping with the one-on-one tomorrow. Before I move forward, I want to mention that the slides for this talk are at this URL, bit.ly/nwb-dandy-unam. You don't have to go there, you can just follow along, but there are links here to other resources that you might find helpful, or if you want to kind of go back to a slide. So these are fully open slides, they're right there. If you just click that, you'll have access to them. Yeah, so if you'd like to follow along, just go ahead and do that. I just want to go over the agenda really quickly before we move forward with the talk. So we are doing the intro talk right now, and then we'll have a lunch break, and then we'll do a 2-hour more in-depth session. So here we're going to really talk about kind of the ecosystem and an overview of what kind of problems we're solving and how we're solving them. And then in this 2-hour session, we're really going to give you in-depth step-by-step tutorials for specific tools that allow you to convert your data, to publish your data, and to access data on the archive and analyze it. So we'll really walk you through like specific software and coding tools to do that. And then tomorrow, what we're going to do is we're just going to kind of show up to the same space. By the way, this afternoon and tomorrow we'll be at another location in the Instituto de Fisiología Celular. Pardon my Spanish. I'm sorry. I'm trying. I'm learning. So we're just going to do a one-on-one session here. So basically, you come, you bring your data, and it's whatever level of experience you have. If you just need help installing things, or if you already know and you just want to ask us very specific questions, whatever you need, it's to kind of we'll be there to help you tomorrow from 10 to 4 in this room. Or if you want to just kind of discuss strategy of what you would want to share or what you would want to access from the archive, kind of all of the above, we're happy to be here to work with you. Okay, so first I want to discuss the need for open data in neuroscience. So first of all, the data that we collect is very expensive. And I think about this expense in 3 different ways. It's expensive in money, in that the equipment that we use to collect this data is very expensive sometimes, and it costs a lot to house the animals that we need. It's expensive in time. It can take months or years to collect the data, and it also takes a lot of time from highly trained people, as you all know, to actually perform the data collection. And a third piece is that we often use animal lives when we collect this data. And so we know that it's really our responsibility to make sure that we extract as much value from this data as we possibly can. And we know that many of the data sets we collect, while they're useful for the specific scientific question that we were exploring, they are also useful for many other scientific questions, right? It could be used by other groups to answer other things. But the problem is that sharing neurophysiology data can be really difficult. And I came across this problem in my PhD when I wanted to work with other labs, and they said, oh, let me look at your data. And I sent it to them, and they said, I don't know how to open these files. And then I tell them how to open it, and they're like, well, I don't know how to interpret these files. Like, what electrodes were recorded from and what parts of the brain. I don't understand this nomenclature. It would take months of back and forth communication. And that kind of seems crazy until you try to do it. And it really is months of back and forth communication. And it can be incredibly tedious. Often sharing data even within a lab can be difficult. I mean, we've talked to so many labs, and they say, oh yeah, this postdoc collected this data 2 years ago, and we want to analyze it, but we don't know how. Like within a lab. And if that happened to you, don't feel bad. We've talked to dozens of labs that have told us this, right? So it's a very common problem. And the main reasons why it's so difficult is that there is incredible diversity in the different formats, right? Because almost every different acquisition system records data in a different format. The data is very high volume, and sometimes it can be incomplete. Sometimes there's a sampling frequency that's just kind of common knowledge in the lab, and it's not necessarily included as metadata in the data set, something like that, right? Or certain nomenclature about brain areas that's common in the lab, but it's not common across the field. So we're trying to kind of identify and fix these issues to enable collaboration across labs. On top of sharing data, it's also often difficult to share scientific software for processing or analyzing or visualizing data sets, because it often requires converting your data from one esoteric format to another in order to load it into the software. And so because of this, it's actually often easier for labs to collect their own data and build their own software from scratch rather than collaborating. And so you see lots of similar data sets being collected and lots of similar tools being built. And And the truth is that we think this is an enormous waste in the field. And it's more than just saving the resources of collecting the data again. It's also enabling labs to share data and software will increase the quality of the data and the software and increase reproducibility of science. And I think the inability to collaborate between groups is actually one of the biggest roadblocks that we have right now, one of the biggest blockers to scientific progress. And if we're able to easily facilitate exchange of data and exchange of software and working collaboratively on these, I think that will have a huge effect on increasing the velocity of science and in understanding of the brain and in finding cures for neurological diseases. So we're envisioning a future where using open data is the norm, where if you have an idea for a scientific question, currently most neuroscientists, when they have a new idea for a scientific question, they first think, what data can I collect to answer this question? I want to change that to, what query can I run on an archive to see what data exists already, to see if maybe that can be used. Maybe it's not perfect to answer the question, but it can be used as a starting point, and it can help me understand what more data I need to collect. When a neuroscientist writes a grant, they talk about what data they need to collect. It's pretty rare, I think. I mean, I don't have access to reading all of the grants, but from what I've seen, it's rare for neuroscientists to talk about what open data exists. But I want that to be the norm. I want that to be the expectation that if you write a grant, you talk about what data already exists and you justify any collection of data in the context of what data already exists and is already open and you can use. So that's the vision, right? Is that the use of open data is integral to the entire scientific process. Now, one thing that's really helped us move this along is this new policy from NIH. Now, I'm used to giving this talk in the US where everyone's very familiar with this rule, but here, maybe not. But so I'll let you know that this recently rolled out as a rule from NIH for all NIH-funded projects starting in January of 2023. They had this new rule that says, if you publish a paper, you have to also publish the data, publicly release the data that was used to write that paper. And if you have a grant and you never publish a paper from that grant, then at the end of the grant period, then you have to publicly release all of the data that you've collected over the course of that grant. Now, what counts as data that you release is a bit up for interpretation. I mean, there's a lot of data that we collect that's like, oh, the system was misconfigured, right? So it's actually junk data. And so you don't always have to share every single piece of data that you collect. But the idea is, look, this is publicly funded research by American taxpayers if it's NIH funded. This is like the United States NIH. And so we should all have access to it, right? All scientists should have access to it. It shouldn't be locked into a specific lab that collected that data. Now, as part of this policy, labs also have to write a data management and sharing plan that details exactly how they're going to standardize and share their data. But the truth is that this is a lot of work, right? And it's something that is a new requirement that many labs don't have experience doing. And this is something that we specialize in. So I'm the founder of a company called Catalyst Neuro. We're an international team of research software engineers that really bridge the gap between software engineering and neurophysiology. And we connect data producers, tool builders, and data users in neurophysiology research. So in terms of what I'm talking about neurophysiology data, what I mean is information recorded from a functioning nervous system, particularly the brain. It include also stimuli and behavior of the animals. And this is often used to study sensory processing, behavior, cognition, or diseases like Alzheimer's and Parkinson's. And I'm showing here on the right, two of the more common modalities. This would be calcium imaging And this would be electrophysiology. And so when we're talking about this data, we're not just talking about the raw data, but here I'm showing a new popular kind of state-of-the-art device probe for electrophysiology, where you would collect these voltage traces from the brain, and then you would often do this processing step called spike sorting, where you're extracting the individual spike times from that raw voltage signal. Similarly, in calcium imaging, you have the raw images collected from the microscope, and then you would often perform image segmentation. This is dead, that's okay. You would often perform image segmentation so you can identify specific cells and the fluorescence traces of those cells. And then increasingly, we're using behavior more and more. So you have the raw videos, and then you also have these new automated pose estimation methods. So the point of this slide, the point I want to make is that when we're sharing data, we need to share the raw data. So the raw voltages or microscopic images or videos, but then in just about every neurophysiology modality, there's some processing step that you need to do in order to actually get to the neurophysiologically relevant signal that you want to actually analyze, right? So we want to share that as well. So this is challenging for three main reasons. One is there's a huge diversity of data formats. Two is that the volume of this data is huge. It can be at the terabyte scale. And three is that, oh, thank you so much. Perfect. Appreciate it. Three is that these experiments can be very complex. So you're often recording from multiple different acquisition systems simultaneously. So maybe you'll have a electrophysiology signal system, and then you also have some kind of behavioral apparatus screen, and then you need to do some kind of synchronization by sending signals back and forth. So that's often very custom. It makes it very difficult to share data with other groups because everyone has a different way of doing this. So to solve this problem, we're on two teams. So we're part of a larger effort here. So I don't want you to think that it's just a Catalyst Neuro thing, but we're part of two big projects to try to solve this problem. The first is Neurodata Without Borders. And this is a project led by Oliver Rubel at Lawrence Berkeley National Lab. And this is a data standard for neurophysiology and behavioral data. And then the second project is the DANDY Archive, which is led by Satra Ghosh and Yaroslav Helchenko. Satra's at MIT and Yarek is at Dartmouth. And this is an archive and a collaboration space for sharing and analyzing neurophysiology data. So the way I kind of like to explain this in simplest terms is that NWB is kind of like the MP4 data format. So I don't know if you all remember, like maybe 20 years ago, if you had to download a video, there were like 20 or 30 different video formats, and it was really hard to like find software that was able to play a video, and it was a mess. Now, videos are almost always MP4 or one of a few other formats. It's really easy. You know, if you send someone an MP4 file, they're going to have the software that knows how to read it. It packages all the data in a way that is universal. And it is a very performant data format. It has high performance because it has special data compression methods that work well for videos, right? So it has universal readability and is able to be very efficient. And then I think, so that's what I think of as kind of where NWB or Neurodata Without Borders is kind of like the MP4 for neurophysiology data. And then Dandy, I kind of think of like YouTube. Anybody can submit data for free and host it there. You can search for it, you can identify relevant information, and you can download it, you can stream it, you can use it, incorporate it into your projects. So we're trying to make this NWB data publicly useful through the Dandy archive. So first, NWB. So the goal of NWB, or Neurodata Without Borders, is to package all of the metadata package all of the neurophysiology data with the metadata that's necessary for analysis. So this, of course, includes the recorded data itself, right? It includes the voltage traces from your extracellular or intracellular electrophysiology or optical physiology, such as calcium imaging or fiber photometry increasingly. But it also has information about your data acquisition system, your experiment design, your subjects, and the behavior of those subjects. It also has a protocol for building extensions. So neurophysiology is constantly changing, evolving, new acquisition systems, new analysis programs, new libraries are coming up all the time. And we can't simply if we try to standardize everything today, tomorrow it would be out of date, right? Because there would be some new system that we're not able to handle. So this extension library allows you to build upon the core schema and allows you to provide new types of annotations, new types of data, where you can handle any type of new sort of data. You just need to build the extension. And this is something that Alberto will go a little bit more in depth into later today. So I just want to give you an idea of what an NWB file looks like. So they're built in this hierarchical way where a single NWB file holds all of the information for one session of an experiment. And you can have metadata about the species, such as the subject of the species, who ran the experiment. And you can have also metadata about, for instance, if you're doing extracellular electrophysiology, you can talk about the electrodes and where they exist in the brain. And then once you have metadata about those devices and the specifics of the experiment, then you can add the actual recorded data. So here I'm showing, this is where you would put like the voltage traces from an electrophysiology experiment. And you can specify the data itself and also descriptions of the data, conversion factor. You can store it with a sampling rate or with individual timestamps if you have irregularly sampled data. And then this electrodes would link back to the electrodes table, so you would know where all of the electrodes were for each of the channels. And then once you have that, you can also store the result of process data. So this would be a units table where you could store all of the results of spike sorting, and also information about each of the detected units, such as the waveform mean, standard deviation, what electrodes it came from. So that's just one kind of glimpse into the schema. We define data types for lots of different types of common structures in neurophysiology, including extracellular electrophysiology, intracellular electrophysiology, optical physiology, behavior, stimuli, experimental metadata. And at its heart, really, NWB is a list of requirements. So if you want to store electrophysiology data, you also have to store what the sampling rate was and what the electrodes were. If you want to store 2-photon imaging series, then you have to provide information about what region of the brain you recorded this information from. So it's really a system of requirements of precisely what metadata you need to include and then where to include it so that we can standardize across different experiments. So this is the whole NWB ecosystem. So there's the standard itself, and then there's software that we maintain around this standard, which includes software for automatic conversion, inspection of these files, ways to read and write it in different programming languages, and software for building and managing extensions, and then software for managing the schema itself. And then we work with a lot of analysis, visualization, and processing tools. It's really a big part of our project to try to integrate with some of the state-of-the-art tools. So we have tools for data exploration, tools for processing of extracellular electrophysiology, calcium imaging, behavior, intracellular electrophysiology, data management, publication, and analysis tools. I'm going to walk through just a few of these tools, but if you want more in-depth information, we maintain a running list of all of the software that we integrate with. So if you go here to nwb-overview.readthedocs.io, we have a list of all of the software that we maintain, and then also a list of all of the software that integrates with the NWB standard. that allows you, all of the software reads and or writes NWB files. So if I've convinced you that NWB is perhaps useful, then you might be interested in converting your data to NWB, understanding how you would actually do that. So we have three kind of tiers of ways of converting data to NWB. The first is the what we call the NWB guide, which stands for Graphical User Interface for Data Entry. This is a no-code application that you can download onto your computer, double-click it, and tell it where your data is, and it will help you download it, or it will help you convert it and upload it to Dandy. Then we have NeuroConv, where we've tried to automate as much as we can. It still requires a bit of programming, but with just a few lines of code, you can convert your data to NWB. And then we have PyNWB and MatNWB, which are interfaces in Python and MATLAB and provide the most customization, but also require a lot of programming. So this is what the guide generally looks like. So you'll open it up. It's a simple interface for converting data to NWB. It supports over 40 different formats, such as, Spike GLX, BlackRock, Plexon, lots of the major manufacturers and file formats that we've seen. We've worked now with over 50 labs building conversion pipelines. So through that, we've managed to see lots of different data files. We supported as many as we can here in the software. This is also, I should mention, backed by NeuroConv, which is the Python library that automates the conversion. So if you need a little more flexibility, you can use NeuroConv and you'll have the same capabilities. So what you'll do here in guide is you'll say what data, what types of data you want to convert, and you'll say where that data is on your computer. And then guide will load in those files, extract as much metadata as it can from those source files, and then present you with a form where you can fill in any missing metadata. So what we found is that we can extract a lot of useful metadata from these files, but it varies a lot. And there's almost always missing metadata that's required by NWB and Dandy. In particular, subject-specific data. So Dandy wants to know what's the species that you're recording from, what is the age, what is the sex of that species, or what is the sex of that organism. And that's very rarely in the source data files that you would output from like BlackRock or from like TDT or whatever recording systems you're using. So this is an opportunity for you to add that information. Then you would, we have interfaces where you can automatically visualize the data, make sure that it was converted correctly. And then finally, we provide an interface for you to either create a new data set on Dandy or to use an existing data set and it will just automatically upload it, and then it'll be publicly available to anyone, just like YouTube, right? And you don't even need to have an account on Dandy to access this data. Anybody could go. If you just go to dandyarchive.org, you'll see hundreds of data sets. So the next step, the next kind of tier of conversion is NeuroConv. This supports over 44 different data formats in electrophysiology, in optical physiology, in behavior, both raw data and process data. And here you'll see just a few lines of code where you specify where the data is, you extract the metadata. Here I'm showing how to edit that metadata. So here I'm adding a time zone to the session start time that's been extracted from the file. Like it has a session start time, but it doesn't say what time zone it was in. So I'm adding that in. And then you just run the conversion. You say where you want to save it, and you include this metadata structure. And then, boom, there you go. You'll have an NWB file. So if you're not used to programming in Python, this might be a little foreign to you, but hopefully you can kind of see the logic of the code. And it's really just a few lines of code. And then if your format is supported by NeuroConv, you're good to go. And I should say this automatically optimizes for archive cloud storage. So it will automatically apply state-of-the-art chunking and compression. So this will make it really performant for analysis and visualization tools to read that data directly from an S3 bucket, which is where it's stored. And then finally, you have PyNWB and MatNWB. This would be necessary if you have really custom data. So a lot of the times we've seen labs where they have their own kind of behavioral setup, and they record that in their own custom CSV or Excel file or some text file that's unique to their lab. NeuroConf is not going to know how to deal with that, right? So you're going to have to write a bit of custom code in Python or MATLAB to add that to your NWB files. This is really useful for trial information, responses, and other properties of the experiment that are not included in the source data. It also this is where you would use if you want to build an extension. There are lots of documentation and tutorials. We're going to go more in depth into that into the afternoon, but we have dozens of tutorials for all different types of modalities in PyNWB and in MatNWB. And so the goal here is you wouldn't write these from scratch. You would copy over one of the tutorials and then integrate your data in, and hopefully it won't take too many lines of code to do that kind of thing. Okay, now I want to talk about Dandy, which is an archive and collaboration space for neurophysiology projects. It includes electrophysiology, optical physiology, behavior, and images from immunostaining experiments. This screen grab is actually a little old. We have many more than this number of data sets and size at this point. Okay, so the goal of Dandy is to store neurophysiology data and associated behavioral data. If you're storing neurophysiology data, it currently requires the NWB format. It will validate those NWB files. It'll check those NWB files for best practices, and it will automatically extract key metadata from those NWB files, which allows you to efficiently search the archive. So for instance, your NWB files will say, oh, I recorded from a rhesus monkey, or I recorded from a mouse. The Dandy archive will extract that information and will allow a user to search the archive for specific species, for instance. So one of the most incredible things about the Dandy archive is that you can upload hundreds of terabytes of data for free. And I know that sounds a little too good to be true, but I'm telling you, we've been doing this for years and it's worked out. So this is made possible by this partnership with AWS, their open data sponsorship program. So a few years back, AWS said that we really want to be supportive of open science, and they have this program where archives can apply to use this program, and which allows them to upload data, download data totally for free on standard S3 buckets with standard S3 performance. So the amazing thing about this is big enough to handle all raw data from most neurophysiology experiments. And so, previously, I think a lot of labs were thinking when they wanted to share data, they're like really thinking, oh, well, what's the most useful part? I certainly couldn't share all of the raw data, so I'll just share the process data. But now that is not a constraint. You really can share both. And that's really great because at least in electrophysiology, I know spike sorting is not a fully solved problem. Like we don't think, when we get our results from electrophysiology, we know that like this is just the best we were able to do given the technology of the time. And we know that we're missing things, right? And we know about specific shortcomings of these algorithms. They're just difficult to solve. So by sharing the raw data, you enable somebody to try to develop a new algorithm and see if they can get better results. And also by sharing the process data, you're able to enable a user to access that data and get running with actual hypothesis tests right away, right? If they want to trust your spike sorting, then they can do that. And then they can, compare one condition versus another on day one. So that's why I really like to encourage labs to share both the raw and the process data. So you can upload to this, you can use the guide as I just showed you, or you can do this programmatically, and Alberto will show later today how to do this programmatically. So this started back in 2020, and since then we have over 300, almost 350 individual data sets, or we call Dandy sets, on the Dandy archive. Now, this axis is actually for both of these lines, because coincidentally, they're almost equivalent. The number of total terabytes of open NWB neurophysiology data on the archive and the number of data sets themselves. They're both at about 330 right now. Many of these data sets, I don't know if it's most, but maybe about half of the data sets are for mouse. We also have rat, human, monkey, fruit fly. We have some cattle data sets, C. elegans, zebrafish. So many of the standard model organisms that you would expect, and also lots of other species. I mean, we have one on ants, which was pretty cool. So like all sorts of stuff. And we're really open to different things. So, you know, you do see a prevalence of the standard model species. Now, these data sets, they range from tens of megabytes to tens of terabytes. Not to say that a large data set is necessarily more influential, but we have been able to provide the storage service for large data sets that would be difficult to share otherwise and expensive to share otherwise. And so the modalities, the most popular is extracellular electrophysiology. We also have a good number of optical physiology, mostly calcium imaging and fibrophotometry. And we have some intracellular electrophysiology as well. We have about 30 of those. Now, one of the biggest data sets that we've worked with, I want to just highlight a few of them. One is the International Brain Lab Brain-Wide Map. We house all of the electrophysiology, eye tracking, and behavioral monitoring for their entire multi-lab collaboration, which is over 33 terabytes of data freely available on the archive. There's another project that I think is really interesting, the Microns project, which is where they did optical physiology, calcium imaging for the visual area, and then they took that region of the brain out and they did electron microscopy to look at the structural connectivity. So this is a data set where you can actually co-register between the cells identified by calcium imaging and the cells on the structural side, and you can look at the relationship between functional connectivity and structural connectivity on a single neuron resolution, which I think is pretty unique. And I think this is a really valuable data set that is underutilized right now. And then we work closely with the Allen Institute. They have a program called Open Scope, where labs can actually submit proposals for the Allen Institute to use their experiment infrastructure, their very sophisticated methodologies to do your specific configuration of an experiment. So this has been going on for a few years. I've linked a few of these here. I think actually we're up to 7 data sets. So OpenScope, what they'll do is they'll do the experiment and they'll give the proposers of that experiment exclusive access to that data for 1 year. And then after that year, they release it on the Dandy archive openly for everyone. And so now, yeah, we have 7 OpenScope data sets available. These are data sets collected by the Allen Institute and really with the intention of sharing it publicly as a public resource. One of the other really cool resources that has been produced by the OpenScope program is this thing called the OpenScope Data Book, which is an interactive neuroscience tutorial textbook that uses the data collected by OpenScope. So it shows you how to read that data, how to visualize it in a number of different modalities, how to do first order analysis, such as identifying receptive fields. And then if I were able to scroll down, they also have really sophisticated kind of dimensionality or reduction types of notebooks as well. So if you're new to neurophysiology, or if you want to learn about a new type of analysis, I think this is a really great resource because it includes the tutorials of how to do this, but it's also specifically designed to work well with the data that's already been publicly released by the OpenScope program. Okay, so one of the really cool tools that's been built to explore NWB data is called NeuroSift. So NeuroSift allows you to interactively explore NWB data directly in the browser. It doesn't require anything to install. You literally can just access it with a URL, and it streams data directly from the archive. It understands NWB, so it can provide visualizations that are specific to certain neurodata types and access the metadata of those neurodata types. And so this is actually fully integrated with the Dandy archive. So I want to show you a demo of this because I think it really shows off the power of NWB and Dandy and third-party tools. So first is the first data set I want to show you was collected by the Moser Lab. And this is actually one of the data sets that was cited in their Nobel Prize for their identification of grid cells in the entorhinal cortex. So this is simply a data set where you have a rat walking around in a large cage. The position is being tracked and electrophysiological recordings are also being tracked. So I'm just going to show you how you would do this. So you know what, I'm just actually going to go to dandyarchive.org and then I'll just type Moser to show you like really from scratch how you would get here. Oh, perfect. Thank you so much. I'm just going to click this data set. So this is what we call the Dandy set landing page. It gives you information about the data set, a description, a title, the contributors, related resources, which can include the papers associated with it, or can include like notebooks that you've published that show how to read and analyze this data. This is metadata that's been automatically extracted from the NWB file. But what I want to show you here is actually accessing the data itself. So you'll go into Files. I'm just going to click a random subject and session. Let's try this one. Open with NeuroSift. Oh. Oh no. Maybe. It's okay. I'm gonna try VPNing in. A little live debugging. That's always fun. Yeah, that worked. Okay. So I'll select here the electrical series, the units, and the position, time series, and X versus Y, and I'll say view. Okay, so this is automatically streaming the raw data from the Dandy archive, and then NeuroSift understands how to visualize these data streams. So here on the first panel is the time series of the voltage. And then here in the second panel will be the spike times. And this third panel is X versus Y. And on this fourth panel, or X and Y over time. And this on this fourth panel is X versus Y. Okay, perfect. So now we have all of these data streams. And if I click here, you can see this red dot moving around. I can zoom in, zoom out. All of these panels are temporally aligned with each other. You can see the actual spikes from, what is it, 1, 2, 3, 4 different sorted units, and the raw voltage. So all of this is freely available. This is a way for you to interactively explore the data without having to write any code. You can go into a file and see, okay, what's in there? What does the data actually look like? Is this going to be relevant for my research? And again, you know, I can take this URL, and I guess you have to worry about firewall issues, but you can pass this URL to a friend, and they'll be able to access exactly this dashboard. The other demo I wanted to show you is a peristimulus time histogram for the International Brain Lab dataset. So if you're not familiar with this dataset, there's a head-fixed mouse. and they are recording electrophysiology while showing the mouse images on the left and the right with varying levels of contrast. The animal needs to use a wheel to indicate whether the visual stimulus appeared on the left or the right. So I'm going to show this. One of the really cool features of NeuroSift is it's able to automatically generate peristimulus time histograms, PSTHs. Still loading. Okay. Oh, yeah. Does it fit well now? A good discussion to have at the end. Okay. So I'll bring it up. Okay, great. There have been some updates to this. I mean, you can see the individual trials and I'll show the trials versus the raster plot. So if I zoom in here, you can see individual trial times, and then if I scroll down here, the unit should be loading. And there's a visualization in version 1 that I actually want to show you. So I'm going to switch over. We just switched to version 2, which is prettier, but we're still working out the details. But if I switch over to version 1, you should be able to see a PSTH. There we go. All right, so I'll load all of these, and then I'll align it to GoQ and response time, and then I'll group trials by contrast left. And so here you're able to immediately start analyzing the data, right? Now, obviously, if you're actually writing a paper, you're probably going to want to do this yourself, and you're probably going to want to specialize it to some very specific conditions. But this is a really good start, right? And already you can see, okay, for unit 1, it's responding more. So this is, if you're not familiar with PSTHs, basically I'm taking all of the neural activity for each one of these is representing one neuron. And then these are all trials aligned to a specific event in that trial. So here I'm aligning to the GoQ, and here I'm aligning to response time. So here with this cell, you can see a clear response to the GoQ that tends to be stronger for the blue and orange conditions, which you can see here are the higher contrast modes. And if you have lower contrast, you see less of a response. So you can see, I mean, there are hundreds of cells here, and you can really explore their individual properties. This one, I think, I'm always really interested to see cells that decrease their firing rate after a stimulus. So this one shows that. So it's kind of maybe a deviation from what you would think by default. Here's one that is clearly responding to the response as opposed to the GoQ time. So you can see all sorts of different cells with different types of response patterns. And this is, again, streaming the data directly from the archive. And I should also say that this visualization was not built specifically for this data set. Any data set that includes a trials table and spike times, you can do this exact same visualization and without installing anything. Okay, in addition to that, if you want to build your own custom analyses, Dandy provides a tool called Dandy Hub, which is a Jupyter hub that allows you to access compute for free. This is, you can go to hub.dandyarchive.org. You do need to register for Dandy first. So if you haven't done that, we can help you with that later today or tomorrow. That will give you a platform where you can run your own analyses. It has fast access to data on the Dandy archive, even faster than you would likely get from your own system, because this hub is actually being implemented on AWS right next to the data. You can write programs in Python, MATLAB, Julia, R. There's actually a server option that includes a GPU. Please don't train a new LLM or mine Bitcoin, but we do provide GPU instances. We also have a way to share these notebooks. So you can create a notebook that demonstrates reading and analyzing open data, whether it's your own data or data from another lab. And then you can share that notebook with the community through Dandy Hub. So we already have now 25 different example notebooks that demonstrate how to read different data sets. So I'll show you, for instance, this Agile 12 data set has 5 different example notebooks that demonstrate how to read and analyze this data. So if you're interested in kind of exploring open data, one thing I recommend is, well, number one is check out OpenScope, right? The OpenScope data sets and the data book. And number two is check out the data sets that already have example notebooks. It'll show you how to read the data and do basic visualizations. So we're really interested not only in getting data on the archive, but promoting data reuse as well. So we've tracked now 30 different papers that use NWB data that's publicly available on the Dandy archive. And you can see that's growing. Every month it's growing. One challenge we've noticed is that sometimes people use data sets and they don't properly cite the data. Like they'll just say they used it, or they'll put the URL, but they won't actually cite it. And that makes it really difficult for us to track usage of data sets. So if you do use data, please cite it as if it were a paper. That would really help us out a lot. When we were looking at these projects, we noticed there are lots of different ways to use data for open science, or open data for your own science. One is a showcase where you have a new analysis tool. You want to show it off on a lot of different data sets. One is a pilot analysis. You have an experiment in mind, but you want to pilot this analysis on another data set that's similar. A follow-up analysis, if there are any unanswered questions in the conclusion of a paper, you can download that data and try to answer those questions. A confirmative study where this is actually, I think, one of my favorites. This is where you collect your own data, you do your own study, you write a paper, and then at the very end, you try to reproduce those exact same results on open data that you found on the archive. So I think this is fantastic because it really strengthens the paper, right? You can say, oh, this can't be due to some detail of how we collected the data. We're also able to find this on a completely different data set, right? It really strengthens the paper. What's a reviewer going to say, right? If you're able to reproduce these exact same findings. You can do a contrastive analysis. You can say, look, this is true for this brain area or this species or this cell type, but it's not true for this other one. And you can find that other one on the archive. You can do an aggregative analysis where you're taking data from many different sources and putting them together to increase your statistical power. Or you can use this as an educational resource. If you're teaching a class on neurophysiology, incorporate open data from the archive, real data, and teach people not only the analysis method, but also how to incorporate open data into your science. And we have a forum where we're collecting ideas for data reuse. If you have some other ideas, we'd be happy to hear about them. So one of the things we're doing to try to promote data reuse is holding this event called NeuroData Rehack. So this is it's a hackathon for generating new insights from existing neurophysiology data sets through secondary analysis. So the idea is we bring in 20 or 30 interested people, graduate students, postdocs, research assistants, software engineers, really anyone who's analyzing data is eligible to apply. And we have teachers, instructors from NWB, Dandy, we have data contributors, we have developers of analysis tools, where for 5 days, we bring you in. For the first 2 days, we teach you how to search the archive, load in the data, read it. And then the final 3 days is really for you to work on your own independent projects of data reuse. And what we have found is that in those 5 days, people are really able to do real projects with this open data, really understand how to access the data and use it. And we've seen some really impressive projects through this. We do this at Genelia Research Campus, which provides lodging and meals for free. We're also working with the Kavli Foundation, and they are doing this, this Neurodata Discovery Award. So it's a $50,000 grant opportunity that's exclusively available to attendees of Neurodata Rehack. And the idea is to provide funding to enable people to continue on the projects that they started in Neurodata Rehack. So previously, the deadline for this was February 12th. I pushed that out a little bit to allow any of you who want to apply. So the deadline is now Friday for this group. If you'd like to apply, I highly recommend it. It's the only thing you'd have to pay for is travel up to Genelia, and we do also have some limited funds available for supporting travel, which generally is going to be used for international participants. So you have a decent shot of that as well, but it just depends, obviously, on the how many applicants we get from other countries. But yeah, would love to see applications from this group. Okay, so with that, I'd like to thank the support of the NIH Brain Initiative, the Kavli Foundation, Simons Foundation, Michael J. Fox Foundation, the rest of the Dandy and NWB teams. This is like a really huge effort. I'm just here talking about it, but it's teams all over the US, including developers and the executive board of NWB. And I have here a page for resources. We have a help desk for NWB and for Dandy. We have Slacks. We're happy to do one-on-one consultations here in person or over Zoom. We have a newsletter, and we have lots of documentation and tutorials, both in text form, and also we have lots of YouTube videos on our YouTube channel, recordings of talks like this, of previous events, and also like individual tutorials, very specific modalities and programming languages. They're all up there on the YouTube channel. Upcoming events, I think the main one to discuss right now would be Neurodata Rehack, but here's a website where you can find all of our upcoming events and information about previous events. And with that, I'll take questions.
Oh, this is great. No, ¿se oye? ¿Se oye? Hola, hola.
Habla más fuerte.
Hola. So this is great. My question has to do with spike sorting software and how your platform can deal with this. So for example, now we are using KiloSort.
Mm-hmm.
Specifically, in my lab, what we do is we record from we record from 5 or 6 brain areas simultaneously with probes that have different numbers of recording tips.
Mm-hmm.
And then we use KiloSort to do all these spike sorting, as you were saying, depending on the quality of the signal to noise ratio and series of other variables, the quality of the spike sorting can vary from very good with single spikes, they're really beautiful, and other channels where you have multi-unit and borderline with noise. And in my lab, what we are doing is creating databases with clean, single spikes, and then other databases where we have everything.
Yeah.
Just to compare the quality of the analysis. So and many places are trying to solve this spike sorting problem because more and more electrodes are recording more signal, and it's impossible to do it by hand.
Yeah.
When I was a PhD student, I did spike discrimination by hand. And it was very nice, very clean. Now that's impossible. So my question has to do with that, whether, narrow data without borders are, dealing with organizations that are, the state of the art in spike sorting, and where we can use these tools so that our database are, as best as possible to, in terms of spike discrimination.
Yeah.
In our case, using Kilosort is a nightmare because the amount of parameters that you need to use and optimize depending on your recording set, the electors, the blah blah blah. It's really tough. And so I'm wondering whether you're dealing with this, or you're just you have a format that allows you to keep your raw data at 30 kilohertz or whatever, and also the spike times and the shape of the spike times.
Yeah,
that's what you do. But I'm wondering whether also you are talking to the people that are directly dealing with having the best spike sorting.
Yeah. So yeah, well, first of all, it sounds like your data set is really state of the art and highly valuable. I mean, 5 or 6 different brain areas for monkeys. It's like it's really impressive, and we want to make sure that we package this well, right?
Yeah.
So so I guess there's kind of 2 things. One is, okay, would the NWB and Dandy handle this? We do have the ability in NWB to have multiple units tables for a given session. So you could say, like, this is the really processed form, and this is the less processed form, and this is the raw data, right? So then that would allow somebody to go in and choose which level they're interested in working with. In terms of the actual spike sorting methodology, we kind of have actually a parallel effort in helping to develop spike interface, which is an effort to build spike sorting pipelines that are, it kind of makes it easier to build sophisticated spike sorting pipelines, including the pre-processing and the post-processing, and evaluating different spike sorting pipelines, like a lot of machinery around it, the manual curation step. So yeah, we've worked for years. Actually, Alberto was part of this project working with spike interface to make sure that it works well with the state-of-the-art algorithms. The latest version of Kilosort and the previous versions of Kilosort are all supported by spike interface, and spike interface can also read NWB and write NWB.
Okay.
So so that's how we're integrating with the state-of-the-art electrophysiology. It's one thing we've noticed working with labs is that, especially with electrophysiology on non-human primates, you sometimes end up with a lot of motion artifact, and that can really influence the quality of the sorting, right? So that's one of the things we focused on in the last year with spike interface, is implementing state-of-the-art motion correction algorithms. We call it drift correction, but it really has to do with either gradual motion of the probe or kind of it being hit by something. And these spike sorting algorithms, they generally assume that the probe isn't moving, right? So it's just the templates won't move in space, but in reality they do. So Kilosort does have a way of correcting for this, like beforehand, but it doesn't always do a perfect job, especially if you have really dramatic motion artifacts like you do in HPs. So yeah, that's one of the reasons I recommend trying spike interface, because it has newer state-of-the-art tools for managing that. It might make a big difference in your spike sorting results, but without seeing the data, obviously, I don't know if that's specifically a problem. Yeah.
Yeah, and my other question has to do a little bit with the fact that, there is an international effort of trying to identify not only pyramidal cells with, electrophysiologically, but also different types of interneurons.
Yeah.
So you have the typical regular spiking versus fast spiking, clustering with using spontaneous activity and the shape of the action potential. But now that we have these, highly dense electrodes, there are some electrophysiological fingerprints that you can use to go above fast spiking, try to go and identify BIP interneurons or CCK interneurons in behaving. And that's happening. I mean, it's as we speak. And I'm still not sure, we have a final answer about that. But your database platform could handle this type of.
Yeah, so I mean, it's a good example of a type of thing where like we're not necessarily trying to build the tools that would do that classification, but we're more trying to integrate with those tools and then be able to to store it in a standardized way that can be generalizable across tools. So like in terms of identifying cell type by things like the shape of the waveform, I know the Buzaki lab is like really pioneering in that effort, right?
Yeah, there is already
and they have software called Cell Explorer that is intended specifically for that, right? It's one of the main features of that. And that's integrated with NWB, right? So it can read NWB and it can write its results into NWB.
Okay.
And so that's exactly the type of thing. And if there are other people working on even more sophisticated methods, as I imagine there are, then we would love to work with them and see if we can read and write NWB files. There's just too many processing things for us to. We don't want to compete with those tools, right? We want to integrate with them.
Okay, sure. Okay, yeah, understand. Okay, and then I have a third question. You have to exactly with sharing the data.
Yeah.
Sometimes I'm working with a modeler. Well, in fact, I'm working with a modeler. He's in Germany, and we are just exactly having the problem that our database format is not very clear. I mean, it takes some time for them to understand that. But there is also the issue of space. So these guys want to have LFPs. They want to have spike times. They don't care about the raw data. So I guess you can configure the databases in different ways. So from a very general, database with all the information, you can generate one with a very specific information. That is the one that you are going to share with another researcher.
Yeah, you could do that. So one of the cool things about NWB and the types of file structures that it uses is you can actually read specifically the data that you want from a file. So you could share a lot of data, including the raw data. And then if someone's only interested in the spike times, they can just read the spike times. And even they can read a specific, if they're interested in the voltage data, but they only want the first second, they can just read the first second, right? Directly from the S3 bucket. So I don't necessarily encourage people to package it in different ways. I'm just like, well, just alter the reading pattern so you're only reading the data that you need. But in terms of, also in terms of the working with collaborators, I don't think I mentioned it, but Dandy does have the ability to share data in what we call embargo mode, where only the co-owners of that Dandy set have access to it. So we call it embargo mode instead of something like private mode, because the expectation is that you will open the data once the publication comes out, as per NIH policies. Even if you're not funded by NIH, if you use Dandy, we would expect you to adhere to that policy because that's what we're built to facilitate. And also, we have this free storage space because with open data, we're not just providing free storage for any neuroscientist to just do whatever they want. But it would be useful potentially if you're planning on opening the data once your publication is out, to use this as a way to share data with your colleagues and not worry about the expense of having to host all of this data on your own private S3.
I understand. So since all the data are in Dandy, so different users, the ones that have access to this data, they can just choose what they want to use from it.
Right.
But it has everything.
Yeah.
So you don't need to repackage it because it's already up. That's your point.
Right.
Okay.
Right. Yeah, you can think of it more almost like a database in that sense, where you're not going to download the entire database. You make specific queries and you download specific data.
Right. Cool. Very, very, very nice.
Good bridge for the other question that we have on YouTube by Jose Bregada de la Fuente, also known as Pepe, I think locally. He was mentioning precisely this problem or practice where experimentalists pair with theoreticians, and they have this agreement that, okay, you will have our data, but when you publish your theoretical paper, you will add us as co-authors.
Yeah.
And he was asking if we have, I guess that one, if we have a way of enabling it, which I think probably like the embargo mode will work for that. But what are the practices that we suggest around that? Whether we encourage, we agree?
Yeah. I mean, so I guess there's kind of two questions rolled into one there. One is, how do we work in exclusive relationships with theorists, right? And I think we've covered that. But then the other question is, okay, well, let's say we do publicly release it. What's the relationship there, right? Like, is there an expectation that we will be co-authors of the paper or not, right? So Dandy allows you to include licenses, or it requires that you include licenses to any data that you share. But the licenses are really very broad. So there's only two options right now. One is CC0, which basically is very loose. And then there's CC BY, which basically requires that if you use the data, you must cite it. You must like specifically say that you use this data. I generally encourage neuroscientists to use CC BY. I think if you're going to publish data, generally most people want that data to be cited, at least if there are figures in the new paper that use that data. And it's also just in line with the expectations of science, right? If you wouldn't use someone's data and then not even mention it. We have in practice seen a lot of times when people use open data sets that we'll see like the contributors of that data set as co-authors of the paper. It's not a requirement of Dandy. It is something basically on a case-by-case basis. We generally see kind of the whole point of NWB is that there's so much that the data is packaged so well that you don't actually need to communicate at all with the contributors, right? You get all the information you need from the data itself. But in reality, you generally do at some point need to talk to them about like nuances of the experiment. And it can be very useful to talk to them, right? So we do find that often the original authors become advisors on these follow-on projects and are listed as co-authors. But yeah, we don't have that requirement in Dandy right now. We're still kind of, as a field, I think, establishing what the expectations are for usage of open data.
Yeah, I mean, I think Pepe is really, I mean, he's hitting a very, sensitive point.
Yeah.
Because, we monkey neurophysiology spent 5 years, collecting, training, collecting, the curing the data. Yeah. And you can use the same database with different analytical tools to answer different questions. And, in fact, I have published the same database 10 times, at least. So it means that, yeah, we try to keep our, our data close to us, and we don't want to share them. And if we don't have clear rules about, what's going to happen with the data, that could be a problem.
Yeah.
I was in a monkey neuroimaging, neurophysiology meeting, and this was the most critical issue for sharing data.
Yeah.
So how many papers you want to publish in a database before you open your data? And at least, we monkey people are very, besides that we are very arrogant, we tend to protect our data.
Yeah.
So how do you deal with that?
Yeah, I mean, it's tricky because the NIH requirement is that you are supposed to release your data as you publish the paper, right? Which, in my opinion, is quite aggressive, right? It would have maybe been a little bit more palatable to especially monkey electrophysiologists if it was like, well, after a year you publish the data. But no, the requirement is like at the time. And this can be a challenge, right? And it's a challenge specifically for ensuring that there's credit attribution, and also for not creating a situation that your own lab is potentially scooped by your own data, right? By someone else analyzing your own data. Now, I understand the fear of that. I think it's very unusual for something like that to happen. But it, I get that you've spent years and years on this, and you want to make sure that you protect it. I think, like, my dream for this would be that citation of a data set, like if there was a paper that said we used this other data set as primary data, like this figure came from an open data set, I just would really want for that to be recognized as a very important contribution, right? That it would be more than just simply counted as a citation, right? For grant reviews, for tenure track assessments, for your graduation of your PhD, whatever stage of career you're in, that as a community, we all agree to evaluate that as something that's a really big contribution. Like if you submit a data set and that data set has been used by 10 or 15 papers, that's huge for the field. I think the issue now, to be precise, is that there isn't enough trust that that is the case, right? That maybe you would release a data set and maybe it would be used, but it's unclear right now whether that will really contribute to you feeling like there being real credit there, right? Just based on kind of the inertia of how we've always done science. And I
think it's very complicated to regulate this. I mean,
yeah.
At the end, I mean, because yeah.
But actually, on that point, is there any discussion within the NIH or whatever about like what you said, like defining a new metric or something?
Yeah.
Not citation, not.
There is. I mean, there is discussion on this. So there's this new effort to define a data sharing index.
Okay.
It's like meant to be, instead of like an H index, like they have this idea for an S index, and there's this competition going on right now that's for people to like define this S index. But the whole point of that is to find a way to fairly attribute credit to people who are contributing really important data sets. So we're working on it, but I don't have a solid answer right now.
I guess there is also two things. One is there is a first date where you submitted data. So there is an original source of the data, right? So that kind of protects whatever publishing. I mean, you can prove it is your data, right? Mm-hmm. And second, which was one of my questions, I guess, I bet there is some sort of ways of keeping the data safe. Problems such as, I don't know, energy, an asteroid or whatever, like a backups or whatever, and people not messing with the data. Yeah. It's like, okay, you can see the data, but you cannot mess with that.
Right.
You cannot change. You just maybe download like information from there. There is a protection, right? So whatever is published, maybe from someone doing meta-analysis or whatever, they cannot invent things from that data, right? Because they cannot change that. And the other is like, people will know where that data comes from. If you see a published, like a paper, oh, this is my experiment, right? It's like, I guess there are going to be consequences for that, for like plagiarism, of course, right?
Right.
So since there is some tools for finding plagiarism,
yeah,
I guess
yeah,
there is a way of knowing who is using the data, and maybe telling the one who submitted the data, look, there's people using your data, just for you to know, I guess, right?
Yeah, I mean, I don't think we've ever come across a paper that used an open data set and then played it off as if it was their own data. And I don't think anyone would do that. I think people are happy to say, this is where we got that data from. Often the people who are using open data are advocates of open data, right? They want to really show that they've done this. And it's no problem to just cite the data set, right? Yeah.
Yeah, I mean, I guess there could be tons of different complications with that. Yeah, in fact, you know, graduate students of mine, in collaboration with a friend of mine in the institute, we use a shared database to analyze it, and it's almost published. But it took some time to interact with the guy that uploaded, publicly the database.
Yeah.
And something that was kind of frustrating is that it was an open database of a part of an experiment. And we needed other part of the experiment to make the story complete. And these guys didn't want to share the other part.
Mm-hmm.
So at the end, the story that we are going to publish is not complete, but it's not all. I mean, these guys didn't want to upload the whole thing. I mean, that's an example of a level of complication. And of course, if you upload a database like the one that I was telling you, where we have five areas simultaneously recorded in a task, it's going to take years to analyze that database. I mean, years. Mm-hmm. And of course, one thing that can happen is that it's up. Maybe you're going to compete in time with people that are going to be fast at analyzing the data, and at the end they're going to publish before we do. I don't know. I mean, there are multiple scenarios where.
Yeah, but that's embargo case, right? Like, okay, we have time. I guess it's not like a 1 year embargo, or is it? Like in all cases, not just NIH, for example.
Yeah.
Like we from Mexico submitting data is we have an embargo, but probably it's like, okay, I need 2 years maybe or something like that. No, is that or always is 1 year?
So the
rules, probably Dandy has some rules.
Yeah, so Dandy's following NIH, which says you're supposed to share the data when you publish the paper, which I know is quite
once you publish the paper.
Yeah.
Okay.
Yeah, I mean, I can understand it. Dandy would certainly accept data that you submitted years later, but it's the embargo specifically. They would want it to be adhering to the NIH rules, just so that they're not storing terabytes, hundreds of terabytes of private data with no clear timeline of when it would be public.
Right.
Another thing I would say is, look, if you're skeptical about sharing your own data, which I understand, another way you can use the archive is to use other people's data. I mean, we have some great non-human primate electrophysiology data sets, particularly from the Chinoy lab, that we were able to work with their lab to get this data, package it in a way that it's usable, and that it can be part of projects moving forward. So happy to discuss using the archive in the other way, accessing other people's data.
Okay.
I think we need to do the lunch time.
Okay.
Okay, thank you very much.
Yes, thank you
all.
very
much. Thank you.
We'll meet back at 1 in the Instituto de... Oh, in here?
Here.
We're meeting back at 1 here? Great.
1 p.m. Come back here.
Okay, perfect.